Thanks everyone for hanging out for the last day, especially when it's so nice outside. And I say this as a person coming from Boston. I'm pretty sad I have to go back. So I've been thinking a lot this week of like how I was going to phrase this talk. There have been really cool math innovations that have happened. And so I thought as a last talk, I maybe would scale back on the math and really focus on some of the opportunities that exist in this space, things that I've been thinking about. Watching everyone's talks about agent-based methods, some of the cool immune dosing type of modeling, thinking about how these could be used as priors in some of the AI foundations that we're using and utilizing, I think is a gap where I think people in this room could help contribute to the stuff that we're doing. So I thought I'd maybe lay out some of the lessons that we've learned and think about ways as a discussion about where works that have been presented throughout the week could be integrated in stuff that we're doing. Cool. So to get to address the elephant in the room, some people are like, why is Microsoft research at a thing like this? So I co-run this thing called Project Ex Vivo, which is a collaborative effort between Microsoft, the Broad Institute, and Dana-Farber. It's co-run by myself, Abba Amini, who's actually technically trained as a biophysicist, but does a lot of computation in AI, Peter Winter and Sri Raghavan. And we're a collection of people both in the computational sciences, anything from pure math to statistics to computer science, all the way over to wet lab biologists, where we actually have a physical wet lab space. So just no, I'm a statistician by training. I have my PhD in statistics. Actually, I got my PhD from Duke, so things that you see are going to be maybe coldly Bayesian, sorry, about the way I think about stuff. For those who don't know, Microsoft Research is located in Kendall Square. So we are here. The Broad Institute and our wet lab is actually right down the street. And we get actual inpatient samples that we get from Dana-Farber across the river. From those who haven't been to Boston, this looks like it's really close, but that river actually makes things kind of far. So that's our kind of group. We focus on primarily two basic themes in our research. The first is this idea of thinking about the fidelity of cancer cell states, and I'll kind of talk about this in a second. But one aim is very biological. We ask, how does the environment affect cell state and our ability to understand therapeutic response? And the second aims are really computational. So really we think about building foundational AI models and this way of building what I'm going to call a state to function map, which I'll describe in a little bit. Really with this aim of, can we learn like a foundational AI model of cell state? Okay, so those are going to be the two things. For this audience, of course, I'm going to focus on this latter piece. So what do I mean by cell state? So our group really thinks about things about moving beyond the DNA landscape that we are currently in when it comes to everything around cancer, right? And so we work on things that we determine state to be a mixture of things happening in DNA, epigenetics, interactions with your environment. And we use as a proxy this notion of RNA to approximate what we consider to be cell state. And so cell state to us, as I'm going to define this in this talk, is going to be like a group of genes that kind of are co-expressed by a cell, and we're going to measure that via RNA. This idea of it captures both cell function as well as some notion of environmental context. So our largely the way we think about drug discovery pipelines, experimental pipelines, are all really centered around DNA, right? You think about a panel of mutations, right? You develop both in vivo, develop ex vivo-like models, right, based on recapitulating DNA markers that you might see in a patient, right? And the way that you enroll people on therapeutic clinical trials is also based on DNA. Right? Do you have the same matched mutations that matched what I had in the clinical trial? Then you get enrolled on a therapeutic, right? What we argue in a couple of papers, and it's been shown really well, is that we all can have the same DNA mutations, which are actually kind of interesting, but actually have different cell states that are represented, right? And a classic example of this is in pancreatic cancer, where there's two classical cells, there are two common cell states, classical and basal, where one in the classical setting is more sensitive to chemotherapy, but more likely to suffer from resistance, right? In the basal state, you actually have a high fidelity of having effectiveness with KRAS inhibitors, right? And these actually have different types of prognostics that are involved. So in ex vivo, what we want to kind of do is that the idea of using state as a way to search for a few things, right? We've used state to kind of show in a Cell paper that if you recapitulate state, your ex vivo models will better align with in vivo responses. We use state as a biomarker, right? This notion that you should use state as a notion for when you enroll patients on certain therapeutics. And our real goal is really to think about state as a notion of actually a drug target. That's our dream, right? And so, this idea of like, how do we kind of measure complex cell state attributes? Now, in this notion to do this, we really want to use AI as an ability to kind of search through search spaces to identify, biomarkers and signatures of these states. They do this kind of like in silico screens for new drug therapies. And so we're going to do in this talk, we're going to leverage a lot of single cell data, right? And I think a lot of people have talked about this. The notion I really love about single cell is a few things. One is currently there's so many different resources now that exist where you can start to have single cell data that are publicly available at scale, right? And so you think about notions like the CellX gene from CZI, right? Publicly available resource. Tahoe Therapeutics has been putting out large corpuses of perturbations, right? And so what's really nice about single cell is both equal parts of computation as well as biology, which is really interesting. So, my group has been working on this notion of foundation models. And I'm going to go through a series of things that we've observed in working with these models. It's going to sound very negative, but I want you to think about this as a part for, a call for opportunity, right? We're not there yet in terms of AI, but I think a notion of giving everyone a roadmap of what we've seen maybe will open up some cool places for maybe collaboration, or also places for you all to take to your own labs to think about how to improve these works. So what do I mean by foundation model? So a foundation model typically is this notion of I take a data set, I feed it into a model, and I typically want to do one of two general tasks, right? And I'll lay them out here. We'll start with the bottom one. The first is the idea of a zero-shot task. In zero-shot, what I mean is I take a model, or take a data set, I train a model, and I freeze that model and I apply it to things downstream. Without touching the weights or anything, I apply it to my given study. So that's a zero-shot case. Fine-tuning means I have a target that I really want to apply my model to. I'll take a model that was trained on some other data set. I will freeze those weights, and I'll take a small amount of data from my target distribution. I will retrain my model a little bit, update those weights to kind of be in line with the target, and then I'll apply it downstream for prediction, okay? And so you can imagine what makes a foundation, what makes a model foundational, but you can imagine there are really cool scientific application areas where a given task might fall into both the fine-tune or zero-shot discovery regime.
So can I ask you quickly?
Yeah.
When you fine-tune the model, do you freeze all the layers except for the last one and only tune the last one, or do you tune all the parameters?
Right, so here we go. Okay, this is a great question. So typically the easiest way to do it is you just freeze the last layer, or you unfreeze the last layer, you kind of update it a little bit with a little bit, right? You kind of like tune the embeddings a little bit for that thing. There have now been studies of asking a few different questions where we call now post-training, which is this notion of like, it's not exactly fine-tuning, but you basically retrain all of the model with a little bit of data from the new thing, right? So I now take a data set that might be of higher quality, or the case might be, than the data set I trained it on. So I retrain the model just on this fine data set, as in what we call a post-training regime, and then I do downstream predictions. Now people started to mix in a little bit of reinforcement learning, which is, I kind of want to talk to some people about their mathematical models, where now I take a frozen model, and the way I kind of update the weights are based on some sort of RL task, like maybe I want to say, like, I want to have a certain immune response, so I want the model to now upweight certain things that are happening in an immune system. And that's why I do my fine-tuning in more of like a reward-based type of system. So there are different ways to do fine-tuning. I'm going to be a very simple thing, where I just do the simple fine-tuning where I take data and I'm going to basically retrain all of the weights a little bit with that new data set. I saw another hand somewhere, but I don't know.
I actually had this same question,
okay.
Whether or not you freeze the backbone.
So for those who don't know how these models typically work, right? Mass language modeling is typically how we pre-train these models. So for all people who may not know sequences, let's take a sentence of how like a ChatGPT works. You'll take a sequence or a sentence, like something that says like learning about AI is awesome. Your first step is to tokenize that sequence into individual tokens. The masking part means you mask out part of that sequence, and then you ask the model to basically impute that part. So you feed in the masked pieces, and you ask it to re-impute the pieces that were masked. And then, according to some loss function or some objective, you then determine how good those predictions were. You continue this notion over and over. Okay. Now, in biology, people have tried very similar things. Everyone in this room is going to be like, well, expression is not ordered, I know. But people have tried this in many different ways by just taking the same technologies that are used in like language modeling and applying them to biology. So let's imagine that you have a set of genes and their expression. What you can do in a very simple case is like take parts of this, randomly order your genes and their expression, mask out parts of this sequence, and then ask the model to then play the same game where it then re-imputes, right? And then you ask how good this notion is. Okay. Now, this framing is interesting because there are many areas of biology where these kind of like foundation models have had more success. I'm not going to say they're solved, but they've had more success than others, right? DNA is a really good, interesting case where you have genomic language models. Protein sequences are probably the most famous case because of things like AlphaFold and others that have had a high amount of success. And in RNA, I think we're still trying to catch up. Where we're not exactly sure what the end goal is for a foundation model in RNA. I would argue maybe it's to predict new perturbations of unseen drugs and unseen targets. But there's a notion of we are not in the same regime where these two data sources are, where you have really nice sequence structure, and you also have a lot of data that's available to you, right? We're not necessarily in the notion of a real data-rich regime for RNA expression, nor are we in this kind of sequence type of system with our data sets. But that doesn't stop us from trying. So there are many different foundation models that are out there, and many of them that come online every single day. I saw 5 this morning on Twitter. And every single model claims to be a foundation model, right? So here are a few for examples. I always apologize in case people are listening online. I missed your favorite one or I missed yours. But there are a few themes that kind of exist. One is that everyone claims they're foundational in some sort of way. The second is that they all are trained on these super large corpuses. So GeneFormer is trained on 30 million cells, UCE was trained on 36 million, SCGPT 33, SC Foundation 50. These are actually kind of low numbers as of recently. I've seen models now trained on 115 million cells. And I was at the Broad like 4 months ago, and someone was like, we're going to train a model on 1 billion cells. And I have a lot of questions about why we want to do that, but I'll get into that in a second. And so we really asked this question to begin, why do I actually want to do this, and how good are these models that continue to come online? I can imagine a setting where I've trained a model and I fine-tune it, where the model would be better at that task and doing it at 0 shot. But if a model is truly foundational, right, like a GPT or something like this, you would say, if I haven't actually trained you for that task, how good are you actually doing this kind of downstream things at 0 shot? So we had an intern actually ask this question. So a couple years ago, Kasia, who's now a scientist at the Allen Institute, really asked this question, really 3 things. She did a 0 shot evaluation where she asked, do these models learn the distribution of data well? She asked a second question, which are, do the embeddings capture a desired biological variation? And she asked her third question, which is, do the embeddings correct for undesired technical variation, right? Because you can imagine if a model is doing it well, if I integrate data across different sources, it shouldn't just capture and recapitulate batch effects. Like a very simple thing, right? I should be able to do like integration really well. And so she found some interesting results. So, despite models being, trained on all these data sets, they kind of just predict like the mean effectively. So here's a model from SCGPT, which what SCGPT does is it takes a single cell matrix, it bins the expression values of the single cell into these different bins, and its training task is basically to recapitulate that binning, right? And so if you just work with the gene expression and you try to do predictions with it, what your model ends up doing is it just predicts the mean. And if you work with embeddings, it actually doesn't get that much better. Like you kind of are able to distinguish maybe high bins, but everything else kind of looks effectively the same, right? So not great, but then how does this kind of relate to a model's ability to do performance out of distribution? So we did a really interesting test. So here are a bunch of data sets where we did predictions with. This is mean squared error, so lower is better in our held out training set. What we did is a few different tests where...
Sorry, can I...
Predictions, you use the expression?
Just expression, yeah. So I have a training set and I have a set of unseen cells, and I basically want to predict the prediction of those unseen cells.
Based on what?
Yeah.
What's your input?
Yep, my input is a set of data sets is a set of single cell measurements. So what I've done is I've taken in, so this is like an embedding model. I've trained my model on a set of like held out data, or a set of training data, so training cells. I take that embedding, and then I feed in new data, and I should be able to basically recapitulate the expression of like complete the unseen data set, right? From this generative model. Yeah.
Yeah, I'm very lost. Sorry.
No.
So my query is exactly what?
So your query is I have a set of data from set of single cell data. So I don't know how to draw this, but I have a single. I'm training an auto. You know what an autoencoder is? I train an autoencoder to basically take input data, train to get an embedding, and then I want to basically recapitulate, right, the trained expression, right? That's my training set.
You want to get the identity. You want to get.
Yeah, I basically recapitulate. I have X, and I want X prime to look like X in my model.
And these vectors are expression levels of various genes.
Yeah, they're just counts.
vector
They're just counts. Yep, and each count in my matrix, so I have a set of each matrix is a cell, and so I feed in a bunch of cells, and I reconstruct their gene expression. And I basically want to ask, can I do this now for
things that I didn't see before?
Yeah.
So what you're saying is, if I can just rephrase it, we were told we cannot talk about manifolds, but let's say that we didn't hear that.
Yeah.
So you learn a manifold where all this data was, and now out of this manifold, you don't expect to have that the autoencoder will work, but you're testing whether it does work. In other words, it's a new data on the same manifold. So.
That's a way to think about it. Right. Except here I want to reconstruct the actual expression. So
but the expression is a vector.
It's a vector, right.
The question is that vector is some big ambient space? Is it also in the same manifold?
Yes.
In some sense?
Yes. Here.
The latent space is much smaller in dimension than the.
Yes. Yeah.
But you do this like in the whole like 2,000 genes?
No, you take highly variable genes only. You only do this for like 2,000. You
like
reduce the space first. Yeah. So what they do in single cell is you take like 2,500. I forgot the actual number we took here. We took like 2,500 highly variable genes, and you just do the training and the testing on those.
So how many?
Huh?
How many?
2,500. So 2,500. Yeah.
So really quick, you said that you're doing this actually on the raw counts, not on log transformed or CPM.
It depends on the method. I don't remember. SCGPT takes in raw counts and then it bends the raw.
It does all that stuff.
Yeah, it does all that stuff itself. Yeah.
Yeah. And one thing just to note, HVGs are typically very low expression. So they typically have very low expression levels.
Yeah.
Based on how those are calculated. So have you tried it with other feature selection methods or other sets of genes?
In this work, we tried to follow the same protocols as the models because we weren't trying to build our own better method, although we can get to that in a second. We were just trying to see for what they claim they can do like how good are they actually. And so so now everyone's kind of on the same page. What we do here is in this first thing, what I do is I just take that expression matrix. And my model isn't a model. I just take the mean of every gene, and that's my model. So there's no training. So that's what you get in terms of your mean variable is effectively your model, and then you do your prediction with that thing. The second is if I feed SCGPT random fake data. So I train it now on like not expression, like just randomly sampled numbers that have nothing to do with anything. And you can see how bad the performance gets on that distribution, which that should be the case because I had this huge distributional shift and nothing should correlate anymore. The next are when I train the model on kidney cells only, blood cells only, or forget blood type, we just train it on a bunch of different human cells ranging across different cell types. And what you can see was really interesting is that these models, which are trained on real data, never actually really do that much better than me just taking the mean. And so that was actually really quite interesting. So the next thing we said was like, okay, so maybe out of distribution types of prediction or reconstruction type tasks are really hard for this method. How easier or how high quality maybe are the embeddings coming from these models? So I'll take my models, I'll kind of train them. We first asked, do the embeddings do a really good job of basically separating across cell types? Yeah.
Because if you're like training, say, a kidney and trying to predict on pancreas, that would be the
Yeah. Yeah.
For example.
Yeah. And so we asked, can we train a model on just all of these cells and then ask how well we separate across cell type, right? And so here are the results of this, and I'll kind of walk everyone through it. So here's my actual data on a set of UMaps where we color coded based on different cell types annotated here. If I were to take my data and just project onto a UMAP with highly variable genes, and then do my kind of like clustering, if you will, this is the coloring that you get. There are models that actually do this explicitly, like we'll call integration methods. Harmony is one of them. SCVI is an autoencoder that does this. These are not foundation models, they're very simple. But Harmony does a very good job of clustering data based on their actual cell types, so you get nice separation across these cell types. SCVI, while it's trained on a simple autoencoder, its embeddings naturally do a really good job of having some sort of clustering across cell types. And GeneFormer and SCGPT are actually the foundation models. And what you kind of see is this like actual splattering of all the cell types where we don't have any sort of separation really across these different groups. So our next question when Kasia did this was like, okay, well then what are these embeddings actually learning? And so you could assess these annotations effectively based on sequencing platform. So the platform that each of these cells kind of came from in terms of their assay. And actually, this is what you kind of see. So what you would like to see in theory is that, even in the real data, everything kind of separates by batch. What you like from an integration method to do is kind of integrate across all of these batches, right? And that's what you're kind of seeing, this nice mixing across SCVI and Harmony. What you're actually learning is that GeneForm and SCGP actually just learn batch effects effectively in their embeddings. And so that kind of gave us a reason to pause. And so we started asking these questions. So going back to Kasia's questions, she asked, do these models kind of learn the data distribution well? Like, not exactly, do the embeddings capture biological variation? I put like a maybe there, but not effectively really. And do these embeddings correct for undesired technical variation? And the answer to that was no.
I would still ask again, 2,500 seems like a large number. How do these models perform if you like shrink it further?
Yeah, that's a great question. Oh, we're gonna get there. Yeah. That's a great point. Okay, so I had a question when this happened. So all these groups are like, I had this question of, we're trying to fit these models on like large corpuses of data. I asked this question not from the gene space, but from the cell space, which was, is data abundance or diversity kind of the bottleneck? Effectively, like, are these data diverse enough to really learn all diversity across cells that I really need to know in order to do these tasks really well, right? And so we started to play this game. So this is a recent paper where we asked this question, performance of single cell models, if I took a large corpus of data, so here we have 22 million cells, what we wanted to do was downsample them to effectively ask, like, do these corpuses need to be this large? Or am I kind of saturating in terms of performance? And so, we sample these data sets to 1%, 10%, 25, 50, and 75% of the data, and we did this in 3 different ways, trying to preserve different aspects of diversity across these cell types. The first was uniform random downsampling. The second one was a cell type reweighted, where I know the cell types that are presented. And so I'll downsample by preserving the relative number of these cell types, just having smaller numbers. And the second is geometric sketching, which is effectively an easy idea where we just basically cluster the data and I downsample proportional by preserving the clusters themselves. And so what I'm going to show you is the performance of a model. And what you'd expect to see is that as this number of my training corpus goes up, right, I should see this notion where if 0 is the performance, I should see this notion where performance kind of scales with my data size. And that's not exactly what you see. So you have these large corpuses trained on big data sets, but they kind of tend to saturate at like 1% of the data. In terms of
less than 1%? Because you want to see the curve going up.
You want to see the curve going up.
Yeah.
And it might be worse for 1%. We just didn't do. We had a reviewer ask us, and then we were like, we did so many tests, we don't want to do anymore. So here's what we did as a full-blown experiment. So we took 22 million cells, we downsampled them according to these different downsampling strategies, and then we tested a bunch of different architectures across these downsampling strategies. And we did this for like many different parameters of inputs, all on data sample size, and figured like, where do we go from here? And we asked this kind of question: when does saturation happen, right? So for a given pre-trained model and evaluation data, if I ask my pre-training data set size and I look at evaluation metrics, at what point do I not really learn anything more, right? And the answer to that is like, I don't really need that much data. So here's a zero-shot classification where I want to now predict the cell types of unseen cells. So I train my model I train these different models here in their regular training corpuses, and I ask a classification question, which is like, when you see new cells, can you predict the cell type labels? And what do I need to do that? Well, really, I just need like 1% of the data, in some cases 25, but no more than that to do this really well. This is a zero-shot case. If you're going to fine-tune, you don't really need any data set. It actually gets much easier. If I'm going to fine-tune down the line, just take 1% of that data set. So I need a fraction of these like 22 million cells plus in order to do this quite well. Predicting perturbations is actually more interesting. So here's a setting across these different models where now I want to say I've seen a model, I've seen a bunch of cells unperturbed, and then I want to predict their expression on drug.
Regarding this criticism of taking just not very clear, I don't know if any of these methods that first, I don't remember the one that was recently published by our old staff. I think it's called.
Yeah.
I don't remember. I know the guy that has on the algorithm, and he's been playing with pseudo-val. So once you have your single cell, once you know the cells that you have in your single cell data, what they do is just not necessarily training in billions of them, but just pseudo-val, right? So just sample from the populations that they know, and you don't need to keep sequencing for forever. And actually, you reduce also the sparsity.
Yeah, no, this is good. These are good points. This is going to come up again in like a second as well. Perturbations are actually really interesting, particularly because you're going to find a lot of people fine-tune. So what you do is you train your model on unperturbed data. You see a little bit of perturbed cells. You train your model, fine-tune it on the perturbed cells, and then you ask, okay, for the rest of these cells who I haven't seen, perturbations applied, predict what these perturbations are going to do to these unseen cells. The same story happens. I only need 1% of the data to really do this. What's really interesting is this like non-pre-trained case where this is a model where we don't do anything. We randomly initialize the weights, and then I just fine-tune. And that's kind of all you really need to do, which is like a very interesting thing in terms of like this notion of like, what is a scaling law effectively in this space? We tried a little from some other things some people have said in our revisions. You have this model, but they're kind of small. What if you increase the number of parameters, right? I have a lot of genes, you have a lot of cells, increase the number of parameters, what happens? What's really interesting is here's a magnitude factor of how many extra parameters we add. We cut the model in half, we multiply the number of parameters by 2, by 4. This baseline is if I just took highly variable genes so there's no model happening here. And you can see that these models tend to just saturate in terms of they kind of hit a ceiling in terms of their ability. So this notion of like, I might as well just look at highly variable genes, doesn't have to be 2,500 of them, however many, but they end up being like a pretty good predictor of performance downstream. So this is kind of negative, I know. So the question is like, okay, so where do we kind of go from here? And that's why I think people in this room are quite interesting. So we've been exploring a few different things. One is this notion of, can you kind of change your training objective to be more biologically focused? And is that an ability to kind of improve model performance? So maybe you don't mess with architectures, you just train the model to learn something different. One example of this is this notion of a recent paper that came out where we thought about this for cell type annotation. A very simple task, but a very important one for people in pharma. Where, typically in an atlas, what happens is I have a set of training data. If I split my training data into two groups, right, models tend to have very high performance, but that's kind of, that's misleading. Because really in reality what happens is, in a cell X gene context or an atlas, all of us are contributors. So as our labs finish experiments, data gets uploaded in like a temporal fashion. So really out of distribution means past a certain date, right? I train my model in May, and then everything from June on is out of distribution. I haven't seen those cells yet. When you do it that way, you actually see a huge drop in performance. Models actually don't do that well. Well, cell type annotation is actually a really interesting thing because cells are related to each other, in almost like a dendrogram type of way. But the way we train our models, we don't train them to learn that tree. Here's an example of this. Typically, when we do cell type annotation, you train your model with a loss that's like a flat cross entropy. What that means is, well, if I train my model to learn, just broad labels like a T cell, a B cell, and the labels down the line are from a real immunologist, and they're like, no, that's a specific kind of T cell. Well, I'm going to miss that label because I didn't train my model to learn over the relationship between broad labels and their basically their children.
Really quick, so the labels you're talking about here, these are all unsupervised labels from clustering. So there's no guarantee that any of these things are actually what are being called T cells. There are things that cluster into large groups that are typically extremely heterogeneous. And also, so I guess what I'm saying is that to what extent do these performance criteria reflect actual underlying reality? And to what extent do they actually reflect the fact that our clusters that we generate aren't necessarily particularly reliable to start with?
Right. So I think what you do, and here's a notion that we took, is that you have to give your model, I think, not such a hard cutoff, but I think what people do, right? You're in or out of that cluster, and so that's it, without looking at the relationship maybe of cross clusters. So what we did is this kind of hierarchical cross entropy loss, which I thought was kind of interesting, which is this notion of like learning across the tree. So instead, you teach your model that like, okay, if I assume this to be a B cell or T cell, I also should take into account this idea that there are other types of T cells that this almost like this label probability could be distributed over, right? And if I do that, now my model has this notion of like learning across labels as a tree, and that gives us the ability to kind of increase its performance for out of distribution settings, right? And so we did this across a lot of different things as an objective. And so for certain cell types, you'll do quite well. But for these other cell types, particularly those that are a little bit rare in the population, or maybe those where you don't have so much information richness from, learning their relationships across the tree gives you a huge data boost in terms of your ability to now actually label things and understand those relationships appropriately. This is a call for us to continue to fill out this tree. So we kind of did this really nice thing where we showed that like, for these isolated groups, you don't really learn anything. But as we continue to learn more about the relationships across cells, right? Or maybe you could take this comes from a cell ontology from a biological resource. You can imagine individuals like in this room that also have mathematical models like really starting to fill this out in terms of like your relationship across cells in order to like really improve learning for some of these algorithms. Just things I've been thinking about as I've been listening to people talk this week. So one thing that we really have been thinking about also downstream is the notion of, okay, really, how do we really start to squeeze information out of these models? Well, there's training objectives we could fix, but maybe also there's this notion of like we could really start to come up with ideal sets of training corpora, right? Now, one secret sauce that like OpenAI has for like a ChatGPT is architecture, yes, but it's also like the training data sets that they were trained on, right? A really thing that I think gets underappreciated about large language models is the way that they're trained is about, you want data sets that are both complementary, but also present orthogonal information so that you can kind of learn and basically tessellate across information so that you can now extrapolate out of distribution, right? And so people think about this a lot with concepts, right? How does logic then play nicely with math and other things when they start to build these large language models? You can think about similar ways to do this for biology, where you could say, well, I know the relationship of like cells potentially like in this tree. Maybe the ideal training corpus isn't such that like I get 1 billion cells, is that I have the right amount of cells in my data set. So one thing we started playing with was like, can I start to leverage biologically driven diversity where my ideal data set kind of tessellates across this tree where now I'm like covering the tree with maximum coverage. And so one quick example of like why this matters, let's take a model where I've trained on just blood cells from this data set here. So I have 230,000 blood cells that I'm going to train my model on. Let's take another model where I've trained my model on blood cells, but I also train my model on a different part of the tree that gives me a whole nother lens of biology, where we look at this like what we call this transcription factor differentiation atlas, where we've now gone into these cells and we've done this little ORF experiment I'll talk about a little bit, where we've overexpressed certain transcription factors and driven different states from those transcription factor experiments. Okay. Now, if my out-of-sample test is on blood cells, my blood model is going to do just fine. Right? The transcription factor gives me a little bit of a boost, but my blood model does pretty well in terms of recapitulating the expression matrix of the unseen blood cells. But let's say I want to test on neurons. So I haven't seen neurons at all. Well, the diversity of the blood cells is so low that I have no hope of ever starting to recapitulate what it is that I would get from those to move over to those neurons. And the transcription factor atlas, although this is a very low R squared, still allows me in some way of its diversity, gives me much more of a hope to recover something that just training on just blood doesn't, right? And so we've really been using this as a way to kind of think about ways to build new data sets that give our models a power out of distribution, right? You can ask, does this relate to things that happen out of across other different cells? Yes, you could do this with neurons, kidneys, hearts. It doesn't really matter. This notion that this transcription factor atlas kind of allows me to see more states, allows me to generalize out of distribution better. Okay? So what we've been doing next vivo, as I've put way too many slides here, is to try to think about how we can engineer state heterogeneity for cancer, right, as a lab. And so we've been doing this with actual experiments, where we've been doing these ORF screens, where we take these different ORFs and a set of transcription factors of a library of about 3,500, and we basically overexpress them across different lines in order to push cells to states where I know I'm information poor, right? So this notion of like, if I know that if I want a more classical states or classical state cells, I can take these cells and apply these ORFs and almost drive them to express like a classical cell state marker, right? In order to almost engineer the heterogeneity that my models need in order to perform out of distribution. And so we've been doing this in what we call an ex vivo state to function map, okay? And what we've been doing this is across many different areas, where we take these different types of engineered heterogeneity, whether it be via CRISPR or via these ORFs. We do this across different perturbations, across drugs, across different environments, and we also do this across different tissues, right? And the idea is, we want to build these AI models to basically learn the links across these states and functions. Now, if you're my advisor, if you're my manager at Microsoft, you look at this and you say, this looks like a lot of money, because if I have to fill in every single square here, this is going to take me forever. How many millions of dollars is that going to take me to do? But we've also been learning from our methods that like cell state's a really nice thing, because state is actually preserved across tissues and across contexts. And what I mean by that is you can get a lot of information where if I know a state in context A, it gives me implicitly information about that state in context B. An example of this is in this paper from Roger, where if I train a model on bone marrow, right, adding more of a diverse group doesn't really necessarily give me anything, right? We've done this with like blood cells. So if I train a model on blood cells only, that allows me actually to do prediction in other contexts like quite well, where this blood model actually gives me a lot of information across everything at high fidelity. What this kind of means in summary is that, this looks like I have to fill in every square in order to get all of my information gain, but that's actually not true. There are a lot of settings where if I fit a model with, let's say, a KRAS inhibitor in context A, that gives me a lot of information about if I had fit like this interferon gamma inhibitor or did an interferon gamma perturbation in context B. And so what we've been trying to do is find out models that really allow us to figure out where's the next place that if I do experiment here, I'm going to learn something about experiment here. Or if I do experiment here, it's just never going to allow me to do this, so I need to do the experiment myself in this place. And so those are the places that we've really been thinking about. And to wrap things up, the most natural question that people always ask me after this is, okay, but you're doing all of this in single-cell RNA-seq. What about other modalities, right? I heard epigenetics, which I loved that conversation the other day. I have a lot of colleagues thinking about imaging, right? There are other ways to characterize cell state that doesn't just have to do with just RNA. So how do you think about multimodal modeling? And a lot of people in the field have also been thinking about this, but I also think about this from okay, I have a lot of RNA, and I'm just going to data dump with everything that I also have from other measurements. And I don't think that's also the right way to kind of go about it. You really want to think about the tasks that you're kind of learning. And so one thing that we've really been thinking about are, for what tasks is it useful for me to fuse? Let's say I have an embedding from imaging and I have an embedding from gene expression, and I want to fuse these to have a multimodal embedding for which I do predictions downstream. In what world is it really useful for me to do this fusion? And in what world should I just spend my money on just like getting more data in one modality or the other, right? So here we've been taking like an information theoretic approach to do this, where each modality is going to bring both unique information about that modality, and in a lot of cases they're going to have some redundant information they also have, right? But the key part about multimodal learning is that what you want to do is you want to unlock that is synergistic, the part of the modalities that were not available to you until you brought those two things together, right? And the way that we've been trying to think about this is via like partial information decomposition, where for a multimodal model, you have both pieces that are redundant across those modalities, unique to either one of them, and hopefully this idea that it unlocks some synergistic type of effect. And so in a paper that literally came out, I think on Wednesday, we put out this idea of like a synergistic information score, right? Basically asking what is the relative predictive gain I get from doing multimodal learning versus any one of the best performing unimodal models. And the way that we kind of separate this is thinking about this across different types of fusion-like methods. So we do this across integration, alignment, and aggregation. The simple way to think about this is if I take two embeddings from two different modalities and I aggregate them, I preserve both the redundant parts of them, but also their unique parts. What people tend to do, which I think is not quite appropriate, is they do alignment. So you take your two embeddings from different models and you try to align them. But that kind of kills the unique pieces that each of those individual modalities had. And what you really want to do is integrate them. And so what you want to do is you want to take your different embeddings and somehow do some multimodal learning with them so that you unlock this synergistic effect. But the idea is that not every single task needs this, right? There are a lot of tasks where you can just be in this unimodal sufficient regime. I only need one mode to do this really well. And there's some modes where I really want to think about this cross-modal learning. And so that's the idea. And in this paper, we do a lot of different things where, I just want to show this experiment really quickly and I'll kind of wrap up, is, here's a setting where I train a model on an imaging patch from an H&E slide. And then as I want to predict the cell type expression in neighboring patches, okay? But what you're seeing on this x-axis is me asking that question of predicting cell expression as I slowly have my test patch move away from the patch I learned. And as you can see, doing this task doesn't necessarily benefit from multimodal learning when the patch is really close. I might as well just do things with just gene expression, and that's totally fine. But as I kind of move away, I now am more and more dependent on having synergy across my two modalities. So with that, I'll kind of wrap up. We did a lot of cool experiments here. But effectively, we want to kind of do this kind of lab in a loop thing with our model, where really we build these state to function maps. We triage, and I really like this idea of triaging across genes, which we haven't really done too much on, but really think about this, analyzing our model, interpreting, and then we have a drug arm, which I'm happy to talk about offline. So with that, there are many different people that have been involved, and thanks for hanging out for the very, very last talk, even though it's a little bit different. Appreciate it.